Hi, hello, welcome to my channel. Uh, we're gonna talk about one of the very important protein structure that is actually found inside bacteria, particularly the TB bacteria, or sometimes we term it as Micobacterium tuberculosis. So what you look in here, it's basically the structure, the the actual structure of the bacteria, and then a magnified portion of the bacteria. It's, it's specifically TB bacteria, Micobacterium tuberculosis. Now, within this TB bacteria, we're gonna specifically talk about this particular structure here by the name of ARABINOGALACTAN. Now, we're gonna specifically stress or talk about this particular structure and further explore in depth and know more about this structure here. So this structure is actually found within the bacteria, TB bacteria. So what it does is that it plays some very pivotal or fundamental roles in aiding or helping this particular bacteria. For instance, like it plays a very important in maintaining the stability of the cell wall of the bacteria, and then it also causes resistance and all that. So for now we will dig deep into it and explore more about it. All right, now if we dig deep into the cellular level, and then if we try to examine it or look at it very carefully, we would let's say, for instance, we take bacteria, specifically the cell wall of acid bacilli bacteria, particularly TB bacteria. So let's say, for instance, this is the TB bacteria. Alright, the bluish portion of the TB bacteria outside, it's called capsule. Now, just below the bluish capsule, the blue portion of the TB bacteria, it's called the cell wall. Now, let's further move into the middle part. Moving into the middle part, we have this little tail-like structure. Yeah, as you can see, it's indicated by the greenish color here, and it's just looks similar like a tail of a snake, and it's called flagella. It helps the bacteria to move forward, and it plays a very important role in mobility, making the helping the bacteria to move forward in motion. All right, and then as you can see, we further moving to the bottom of the inner part. If we come here, this greenish space, the space inside here, this is called the periplasmic space, and this is the space between the inner membrane and the outer membrane. All right, so if you basically look at those particularly the gram negative bacteria, they have double membrane, the inner membrane and the outer membrane. But if we look at the gram positive bacteria, they only have the inner membrane, but they don't have the outer membrane. So in this case, it's for Mycobacterium tuberculosis, it's actually gram negative bacteria, so they tend to have the inner membrane and the outer membrane. So the space between the inner membrane and the outer membrane, it's called the periplasmic space. And then the inside the wall towards the cytoplasm, it's called the cell membrane, or sometimes we call the cytoplasmic membrane. Now inside the cytoplasm is actually a liquid portion of the cell. It contains those specialized organelles. All right, so this little purplish pinkish like structure here, it's called ribosome. And then we have the plasmid out there, and then we've got the plasma, the liquid portion of the cell, and then we've also got the soluble RNA, or sometimes we call it ribonucleic acid. All right. Now let's come to the real business. If we take the little portion of this part here, and then if we magnify it and zoom it, we're gonna see a picture like that. We're gonna see a structure similar like this in the TB, or Micobacterium tuberculosis. So let's take this little portion and then we zoom it out, and then here comes the real picture. Now inside this magnified picture, this is the enlarged view of the acid fast bacterial cell wall. Now if you look at it closely, this structure from the top to bottom, from this exact point, it's the cell membrane. Here, this is the cell membrane. This little portion here, just below, towards the cytoplasm. This is the cell membrane. And then if we move further upwards, we'll find what's called peptidoglycan, and this is the structure here. It's just above the cell membrane, peptidoglycan. And then just above the peptidoglycan, we have what's called monophosphinothiols, and these are the little structures that it's actually talking about. And then just next to it, we find the lipoarabinomannan, and this is the yellowish like structure here. It's talk about this one. And then just further moving upward, we found what's called the ARABINOGALACTAN, and this is the main thing that we're gonna talk about it. And then just above it, we've got that mycolic acid. Now let's try and dig deep into the structure of this arabinogalactone, and specifically talk about this structure. Now this diagram is basically the same one that we just seen it previously. All right. Now if we look at it closely in the molecular level, the structure of Micobacterium tuberculosis, it contains this what's called arabinogalactan here, the blue portion here. This is the arabinogalactan. Now this arabinogalactan, it's actually a complex branched polysaccharide linker molecule, meaning to say that it links or it connects two important structures. As we go through, we're gonna look at it. Now what it does is that this arabinogalactan, it actually joins the peptidoglycan, and then we have the peptidoglycan here, and then it joins the peptidoglycan and the what's called the mycolic acid. Now this is the mycolic acid, the reddish color. Now inside this mycobacterium, we found the mycolic acid, and then also we found other structures like acyl tetralose, and we've also find those glycolipids and PDM and all that. So within the membrane, we find the mycolic acid. Now this arabinogalactan, it is highly responsible for joining these two important structures. We call the mycolic acid, the reddish color, and then the peptidoglycan. So it plays a very important there. Now this molecule, it acts as a what's called biochemical bridge. This one, the bluish structure, and it's in an important zone called the medial wall zone. Now this is actually located in this zone. So arabinogalactone. Now it plays a very important and act like a biochemical bridge. Now this structure connects two important layers, like I've said it already. Now, inside the cell wall. Now, this actually inside the cell wall, so it connects this mycolic acid and the peptidoglycan. Now, now the outside, we found the mycolic acid here. Now, these mycolic acids, they are very long chain what's called hydrophobic fatty acids, forming a waxy barrier, meaning to say that hydrophobic fatty acid, meaning to say that they tend to repel water. They have a hydrophobic nature of fat. You can think of it like water and, let's say, fat, or water and oil. Water and oil doesn't diffuse evenly, right? So they separate. So they don't like diffuse evenly. So this mycolic acid, it's actually made up of fats, so they tends to repel water. So that's why they have this nature called hydrophobic. All right, they act as a waxy barrier. Now this galactan, this galactan connect these two layer, and then it maintains the rigidity, or it makes them become very strong, and then it binds them to a bond called covalent bond. And covalent bond is actually a strong chemical bonds that say electrons. They say electrons. Now this bond makes the wall become very integrated and complex, and then it separates, and it doesn't separate these two layers alone, but it maintains the rigidity. It makes them become very strong. This arabino galactan. Why I'm stressing too much on this? Because this structure plays a very important role in clinical aspect, in pharmacology and all that. That's why I need to talk about more on this particular structure and help you understand more. All right. Now, if we look at if you wanna look at it very closely into its biochemical structure of uh, this particular carbohydrates, it is actually made up of carbohydrates. Why I'm saying this? Because they are made up of what's called arabinos. Arabinos, these are carbohydrates. They contain 5 sugars, and then another important carbohydrate or sugar called the galactose. They contain 6 sugar units, and these two branch together, and then they form this arabinogalactone. Now this branching of the sugar units makes them become very complex network, and then this branching increase the structural rigidity. It makes them become very strong, and the cell wall becomes very rigid, and makes it become impermeable. Now, when they become very strong and rigid, it makes the wall become uh, very strong, and then the ability of substance to move in and out of the cell is very low, because it decreases the permeability rate, moving substance in and out of the cell. So substance, like when it comes to gram staining and gram positive, gram staining for positive bacteria and negative bacteria, dyes like crystal violet, it's very difficult for them to diffuse easily through, and then move into the cytoplasm, and finally reach the ribosome and nucleus and all that. Because of this important structure here, they prevent the violet, crystal violet to diffuse into the cell. It's simply because they have this hydrophobic nature, and they tends to repel water. And this crystal violet violet, they are water soluble. So when they dissolve in the blood, and when they reach the mycobacterium and that they want to penetrate through, they tend to uh, become resistible due to this arabinogalactan galactone, because they contain this waxy structure or fatty long fatty tissue, long fatty structures. They they have this hydrophobic nature, so they tends to repel the water. So this water soluble substance, when they want to bypass through, they become difficult, and it stops them, it resists them. So that's one very important thing that we should take into consideration. All right. Now, the rabbinogalactan, it also helps maintain the mycolic acid family and peptidoglycan. Now, without this linker, this lipid layer would gradually loosen, and it wouldn't maintain the rigidity, maintain uh, the rigidity of the cell wall. So the stability would actually collapse. So therefore it's very important that acid fast property, it's actually uh, maintained by this arabinogalactant. Now when the gram staining tries to penetrate, like I've said, is resisted because there is a lipid barrier there. Now in addition to that, the structural enforcement of the arabinogalactan, they also tend to form additional barrier that makes the permeability of substance become impermeable and very difficult. So let's say, even so, when we try to add some little bit of dye, and then if some portion of that dye, if they happen to make it through and bypass through this layer, and if they make it through into the cell, even though the little can pass through, but because of the because of the arabinogalactan, it restrict them because of the dense carbohydrate network of arabinose, 5 carbon and galactose 6 carbon sugar. Now they act as what's called secondary barrier. Now in pharmacology, as in the aspect of pharmacology, like some TB drugs like uh, rifampicin and parazinamide and ethambutol and all that. Let's say, for instance, ethambutol. If this ethambutol, they actually target this structure, arabino galactant. They target this structure basically. Uh, they inhibit the synthesis of arabinogalactant, and then once when they inhibit the synthesis of arabinogalactant, if there's no too much arabinogalactant, then uh, they wouldn't be able to maintain the rigidity of the cell and make the cell wall become very strong, and then they would tend to lose the strength, and then the 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 arabino galactan network structure, the mesh would be gradually decreased, and thus it will loosen it will make the strength of the cell wall to become very what not that strong, and then as a result, it will lead to uh, influx of substance. It will increase the permeability of substance like solutes, or probably some solutes, and then also the water, most importantly water. And then water would tend to move into the cell, and then cell will eventually accumulate so much water in the TB, Mycobacterium tuberculosis, and eventually cause what's called cell lysis, and then kill the TB bacteria. So that that only occurs when this arabinogalactan synthesis is suppressed, or if it's stopped. So in the clinical perspective, it's very important. This structure is very important. That's why I'm stressing about the structure of Mycobacterium tuberculosis quite a lot, because it plays a very important role in clinical when it comes to clinical aspect. Let's say particularly in uh, treatment. Uh, yeah, treatment. So thank you. So this brings us to the end of our discussion, and uh, I'd like to see you on the next channel. All right, thank you.